Thanks for the introduction, Jeremy. So today I'm going to talk about one of the molecules in the dual specificity phosphatase family known as the DUSP10. And as we know that phosphorylation is a major molecular switch of signaling. So when the cell receives a signal, then protein kinase will transfer the gamma phosphoryl group to its substrate. And upon phosphorylation, many of the features of this substrate will be changed, including their activity, the interaction with other proteins, as well as their stability and subcellular localization. So after a while, then the signal needed to be turned off, so the protein kinase will be able to remove this phosphoryl group from the substrate. Therefore, it's very important for our cell signaling. And one example, sorry, I need to go back. So let me see. Previous, yes. Previous, yes. So in human beings, we have about 520 protein kinase, and we only have about 150 phosphatase. And the protein kinase, because of the importance in our cell life, therefore anything mutations will cause a lot of disease. So one such the MAPK kinase signaling pathways are evolutionarily conserved signaling pathway, and whose activation is mediated by a cascade of phosphorylation. So in mammalian species, we have three major groups of MAPK kinase known as ERK, P38, and JNK. And these kinases are downstream target of cell signaling, such as cellular receptors, such as the toll-like receptor, as well as the T cell receptors. So in response to the signaling, and then phosphorylation will from the MAP3K to MAP2K, and eventually lead to the dual phosphorylation of the MAPK kinase in its activation loop. Then this activated MAPK kinase will working together to regulate gene expression, such as the cytokines, then to regulate cellular response, including cell growth, activation, effector function, as well as apoptosis. And we know that activation of these MAPK kinases are important for the expression of the inflammatory cytokines in immune cells, and it is important for antimicrobial infection. However, overproduction of these cytokines will cause pathological outcomes. Therefore, their activation actually is tightly regulated by multiple mechanisms, including the activators, scaffold protein, which assemble signaling complex to mediate the activation, as well as the negative regulator. So not recently, about 10-15 years ago, it was identified that one family of protein known as the MAPK kinase phosphatase, or the dual specificity phosphatase, are the major negative regulator of this MAPK kinase. So then what are these MKPs or the DUSPs? So these DSP or MKPs are system-based protein tyrosine phosphatase. So they are able to actually dephosphorylate both the tyrosine and the trypsin residues in the activation loop of the MAPK kinase. Therefore, they are also known as a dual specificity phosphatase. So originally, they were identified as the major negative regulator of the MAPK kinase. So accumulating evidence that is actually shown that they are also able to regulate the activation of other important molecular molecules, such as STAT5 and FOXO1. So this slide shows the typical structure of this MKP or DUSPs. So you can see here that all the DUSPs actually contains a conserved catalytic domain in their C-terminal. And they also have a D domain in the N-terminal, which mediates the interaction between them and their substrates. And in my lab, actually, we focused on several of these DUSPs or MKPs, including MKP5, also known as the DUSPs, as well as MKP7, MKP2, and MKP3. So today's talk is about the MKP5 or DUSP10. And this molecule actually was identified in the end of last century in both human and mouse. And when overexpressed in cell lines, and it was able to inactivate P38 and JNK. And now we know that this early study have its limitations, probably due to the cell line used or due to the antibodies or technology. So, and about in 2004, we published a paper shows that actually this DUSP10 is a JNK phosphatase in T cells, plays a very important role in both innate and adaptive immunity. More recently, we shows actually this DUSP10 is a RF3 phosphatase is able to inhibit type 1 interferon expression in innate immune cells in response to RNA virus infection. And there are studies shows that actually this the polymorphism, this DUSP10 is associated with CRC, colorectal cancer. However, the function of this molecule in colorectal cancer is not clear. And this slide summarizes our results, our findings on the role of this DUSP10 in innate immune response. We found that in response to toll-like receptor activation or microbial infection, so the signaling will transduce to the MAPK kinase such as JNK to reactivate the transcriptional factors such as AP1 to regulate inflammatory cytokines. On the other hand, so in response to toll-like receptor 4 or other pattern recognition receptors, so the RF3 will also be activated, which will then regulate the type 1 interferon expression. And during this response, so the expression of DUSP10 was induced via an unknown mechanism. This protein was then able to inhibit the activation of JNK as well as RF3 to inhibit the expression of both inflammatory cytokines and the type 1 interferon. And we also found that DUSP10 plays a very important role in adaptive immunity. So on one hand, DUSP10 is required for the activation of T cells. And on the other hand, when these T cells differentiate into Th1 or Th2, then DUSP4 is able to inhibit the effector functions such as the expression of interferon gamma by Th1 or the IL-4 and IL-5 by the Th2. So, the DUSP10 plays a very important role in the immune response, and how it regulates the inflammation in other tissues, such as in the gut, is unknown. So, to understand the function of these molecules in gut inflammation, we fed the mice with the dextran sodium sulfate, also known as the DSS, to induce inflammation, and we examine the expression of these molecules by histology in the gut. As shown here, you can see that the DUSP10 expression is highly induced actually by the feeding of the DSS. So this indicated that this molecule may play a very important role in the inflammation in the gut. So to address this, so we applied the DSS-induced colitis to both wild type and knockout mice. Originally, we expect that due to the inhibitory function of these molecules on inflammatory cytokines, we expect that the knockout mice might develop more severe disease. But surprisingly, we found that actually these knockout mice are more resistant to DSS-induced colitis. So as shown here, you can see that we fed the mice with 2% of DSS in drink water. Then after that, we withdraw the DSS, let the mice to recover, and the body weight loss was recorded. So the red line here is the wild type mice, and the black line is the knockout. The red line is the knockout mice, and the white type is a black line. And you can see that after withdrawal of the DSS, the white type mice start to lose weight, and then they reach the weight loss, the peak of the weight loss at day 9 after the feeding, and then they slowly recover. So in contrast, actually, these knockout mice do not lose their weight, actually, then, which tells us that actually these mice were resistant to the DSS-induced colitis. So we then examine under the histological feature of the wild type knockout mice, as shown in these 2 photos. You can see that in the white type colon, so the architecture of the gut was destroyed. You can see the erosion of the epithelium and also the infiltration of massive inflammatory cells. On the other hand, in the knockout colon, actually, the crypt architecture is much better preserved, and they also have much less inflammatory cell infiltration. And this histological score was presented at the bottom. So this tells us actually the knockout mice somehow are able to keep the architecture of the gut, and to be more resistant to the colitis. So next we examined the berry function of the mice by feeding this wild type knockout mice with dextran in drink water. At the same time, we treat the mice with the DSS to induce inflammation. So as shown here, you can see that in the blood of the wild type mice, so the dextran concentration gradually increased, and it reached the peak as well at the day 9. You can see that there are about 200 nanogram per milligram in the wild type mice, and compared to these wild type mice, actually these knockout mice seems like have better berry function, because they have much lower concentration of dextran in their blood, and this is in line with the increased proliferation of gut epithelial cells at the crypt bottom. You can see here in this Ki67 staining, so which identifies the proliferating cells. You can see that in the knockout column, there are much more Ki67 positive cells compared to the wild type mice. So this tells us actually knockout of the DUSP10 from the mice lead to the increased proliferation of the epithelial cells in response to the damage of the epithelium. And then what is the mechanism? So we took the colon from both the wild type and knockout mice and to examine the MAPK activation, including P38, DNK, and ERK. We only found increased ERK activation in the knockout colon in response to DSS treatment, as shown here. And we also found that a molecule known as KALF5, which is important for epithelial proliferation, the expression also increased, and it has been shown that this is a ERK targeted genes. So this tells us actually DUSP10 is ERK phosphatase in the colon. And in addition, we also found that overexpression, we also found actually DUSP10 are able to interact with ERK, and also they are able to dephosphorylate both ERK1 and ERK2 in vitro dephosphorylation, as shown here. So this established that DUSP10 is ERK phosphatase, actually. So then we know that increased epithelial proliferation is better in terms of inflammation, but we also know that increased uncontrolled epithelial proliferation could lead to the cancer in the gut, is colorectal cancer. And a little bit information about colorectal cancer. So this cancer is the most frequently identified cancer in both man and woman. It is a number 2 cancers in the man and number 3 cancers in women worldwide. And in Singapore, actually, it is a number 1 cancer in both man and woman. So most of the colorectal cancer patients, actually, they suffered the loss of APC, which is a major tumor suppressor in the gut. So the early mutation of this APC actually gives the growth advantage of the developing epithelial cells. And we know that in our gut, epithelial cells will change, will be renewed in about a week, and the new cells will be formed from the crypt. And this developing cell will move up to the epithelium to replace the older cells. However, the mutation of APC and other genes actually provide the growth benefit to the mutant cells, and these cells will then stuck, and then they will form a clump of a cell cluster. But over the time, other mutation will occur, such as the mutation of KRAS or the loss of P53, which eventually lead to the development of carcinoma and invasive as well as metastatic colorectal cancer. So the MAPK signaling pathways, especially the ERK pathways, are highly represented in the cancer. So mutations in this pathway actually often associated with colorectal cancer. For instance, the KRAS mutation actually is identified in more than 30% of the CRC patients. The BRAF mutation, on the other hand, is identified in about 10% of the colorectal cancer patients. And interestingly, the KRAS mutation and the BRAF mutation are mutually exclusive. So therefore, in about 50% of the colorectal cancer patients actually have mutation of this pathway, which lead to the constitutive activation of the ERK to drive the cell proliferation as well as migration. So as DUSP proteins are negative regulator of the ERK pathway as well as other MAPK pathway, so it probably plays a very important role in the development of the colorectal cancer. So to address the function of DUSP10 in CRC, we utilize the mouse model of the CRC. We fed the mice with a chemical known as AOM together with the DSS. So in this model, the DSS will damage the epithelium to introduce inflammation. The AOM actually can cause mutation of the gene in the epithelium. So this mutation together with inflammation drives the formation of the cancer. So as shown here, so the Y type can form the colorectal cancer in the colon. In contrast, the knockout actually forms much more tumors, and these tumors are at more advanced stage. You can see as shown in this histological picture. So in the black here, so in the white type picture, so the small area which is highlighted here actually the entire tumor. So on the other hand, in the knockout colon, actually the entire photo actually is a tumor. And also these tumors are at more advantageous stage as shown in the bar chart. Excuse me. Yeah, in the bar chart. And in addition, the Ki67 staining also shows that the knockout tumor contains more proliferating cells. So together, this data tells us actually the DUSP10 is a tumor suppressor in the gut. So within that is mouse work, and how about a human? So we utilize overexpression actually strategy to examine the function of this DUSP10 in human CRC. So we overexpress the DUSP10 in CRC cell line DLD1 as well as CACO2, then measure the cellular proliferation. As shown here, you can see that the overexpression of DUSP10 is able to suppress the cellular proliferation of these cancer cells. And in addition, we also found that overexpression of DUSP10 inhibits the activation of ERK, as well as ERK-regulated genes, including the KAF5, the c-MYC, and the cyclin D1, which are important for cellular proliferation. So furthermore, we also found that DUSP10 is able to inhibit the cellular migration in this one healing assay, as well as the spheroid formation. So you can see that the DUSP10 overexpressed cell forms much smaller spheroid in this in vitro spheroid formation assay. So in addition, we also inoculated this white vector expressed and DUSP10 expressed colorectal cancer cells into immune deficient mice. And as shown here, you can see that overexpression of these genes greatly inhibited the formation of these colorectal tumors in vivo. So together, this data shows that actually DUSP10 are able to inhibit the human colorectal cancer growth. And how about the patient? So in order to address this, we analyzed the patient survival probability in association with the DUSP10 expression levels. Utilize the two cohort of human CRC patients. In the first cohort, we found that in patients with high ERK expression, if they have a high DUSP10 expression, then their survival probability will be increased about 20%. In the second cohort, we found that it doesn't matter whether patients have high or low ERK expression in their tumor. As long as they have a high DUSP10 expression, then they will have increased survival probability, also about 20%. So this tells us actually indeed that DUSP10 is a tumor suppressor in colorectal cancer in humans. So next, we further confirm the function of this DUSP10 by utilizing these APC deficient mice. As I mentioned, that APC is a major tumor suppressor in the colon, and the function of this APC actually is to assemble a signaling complex which leads to the phosphorylation of one important molecule known as a beta-catenin, and the phosphorylation of this beta-catenin will lead to the degradation of the protein. However, at the conditions where the APC lost its function, then beta-catenin will not be phosphorylated, then it will migrate to the nuclear to regular gene expression, including the expression of these important genes such as cyclin D1, c-MYC, to promote cell proliferation as well as migration. So we crossed these mice with our DUSP10 deficient mice, and we know that APC min mice actually form the small tumors in the small intestine. And as shown here, you can see that after cross with the DUSP10, we found a lot of tumor formed in the colon, as shown in this photo. And also the number of these tumors are greatly induced in the DUSP10 and APC double deficient mice. And this tells us that actually DUSP10 is able to inhibit the genetically mediated colorectal cancer formation. So in summary, we found that inflammation or carcinogen stimulation or the inheritable mutation could cause abnormal epithelial cell growth in the gut which will promote the formation of the colorectal cancer. So in this, in the development of the tumors, ERK activation actually was induced by the mutation of the KRAS or BRAF, which will promote the tumor formation. And if we have the DUSP10 expression during this process, then DUSP10 will be able to inhibit the activation of ERK, thereby to inhibit the formation of colorectal cancer. So that's all that's all for my data presentation, and I would like to thank the people who did this work, and mainly Qingwen here, who wear a red t-shirt, and with the help of the other lab members. In addition, I also want to thank my collaborators who contribute to this project in one way or another, and also my the fundings from NUS or the National Medical Research Council of Singapore or the Ministry of Education of Singapore. So that's all for my presentation. Thank you for your attention, and I will be happy to answer any questions you may have.
Thank you very much indeed. I'll just turn you around so they can see it there.